Hello guys, welcome back to Learning with Isha. Today we begin talking about a very important concept of polygenic traits. But before that, we need to understand two basic terms. First, what is a qualitative trait, and second, what is a quantitative trait? Well, we are used to watching the Mendelian characters. Like height in pea plants, we know two different phenotypes are produced, tall height and short height. So these are two distinct phenotypes produced for a character, that is your height. This represents what we know as a qualitative trait, where one gene is responsible for one character. Simple version है Mendelian inheritance वाला. For example, height of the pea plant, flower color which could be white or purple, all these are examples of qualitative traits. Now, when we come to quantitative traits, what happens is a range of phenotypes is produced. We come across this in common life. Let's say height is one character that is not controlled by only one gene. There are many genes that work together and contribute to the height of the individual. Similar is the case with the weight of the individual. Also, the skin colors in humans. So, a variation in skin color is produced. I cannot say there are two distinct skin colors. Rather, a spectrum of skin colors is observed in humans. So, this is what we know as quantitative trait, also known as metric traits. So, these are quantitative or metric traits. Now, the two words come into play, monogenic inheritance and polygenic inheritance. We know that in qualitative traits, this one, only one gene is responsible. So, therefore, we call it monogenic inheritance. One gene responsible for one trait. Quantitative traits involve several genes. Therefore, it is known as polygenic inheritance. This is exactly the topic for today's video. Let's begin talking about polygenic inheritance. Well, it is also known as multifactorial or quantitative inheritance, as we have already discussed. It represents a situation when one character is controlled by more than one genes. Multiple genes are responsible for a character. Now, these genes that control that character are known as cumulative genes or multiple genes or what we know as polygenes, and thus the name polygenic inheritance. What is special about these genes? The special thing is that none of these genes individually is responsible for the phenotype. It has an additive effect. सब थोड़ा थोड़ा उस phenotype के लिए contribute करते हैं. कोई एक जीन सिंगली उस phenotype को control नहीं कर पाएगी. Let's look at it better with the help of an example. A classic example of polygenic inheritance is the skin color in humans. The skin color in humans is controlled by, let's say, three genes, namely A, B and C. A जीन अकेले स्किन कलर नहीं दे पाएगा, B अकेले नहीं दे पाएगा, C अकेले नहीं दे पाएगा. Together they are going to synthesize melanin and eventually give the skin color that is responsible. Now, if all the three genes are present in a dominant condition, so let's say the situation is A is dominant, B is dominant, C is dominant. The phenotype is going to be of the most dark skin color type. So the melanin synthesized here will be maximum and the skin color is going to be the darkest. Let's say all the genes are of the recessive type. So no melanin is going to be synthesized. And in this color, the skin color is going to be the lightest. And if the situation is intermediate, means all the genes are heterozygous. Here the skin color is going to be intermediate. So now let's move ahead and try to look at this example in detail. So the situation is something like this. We are trying to find out the number of dominant genes because we know जितने ज़्यादा dominant genes होंगे उतना ज़्यादा melanin is going to be synthesized and the skin color is going to be the darkest. The first scenario is an all dominant condition. So in this case, we see the number of dominant alleles is 6, all dominant, A dominant, B dominant, C dominant. So dominant alleles is 6, the skin color is black, it is the darkest. Now we come to an intermediate phenotype. Here we observe a heterozygous condition. The number of dominant alleles is 3. A is dominant, B is dominant and C. So we have 3 dominant alleles in this case. The skin color is going to be intermediate and this is known as mulatto skin color. Now the last scenario is going to be if all my alleles are of the recessive type. In this case, what happens is the skin color is lightest. Why? Because I have 0 dominant alleles. In this case, I am going to have no melanin production. As a result, the skin color is going to be albino. It is going to be very, very fair. Now, we realize that between the 6, 3 and 0, I have scope for a situation of 5 dominant alleles, a situation of 4 dominant alleles, here 2 and 1 dominant allele. So in between the scenario of this 6, 3 and 0 dominant alleles, I can have intermediate scenarios as well. I have a case where I could have 5 dominant alleles. In that case, my skin color is going to become very dark brown. 5 dominant alleles क्या हो सकता है? Let's say it could be a case where I have A dominant, B dominant, B dominant, C dominant, C recessive. Also, it could be A dominant, A in a recessive state, rest of them could be dominant. So in this way, we are not writing one genotype because several combinations are possible. Similarly, if I have 4 dominant alleles, I have a dark brown skin color. 3, I have already said mulatto. 2 would become fair. 1 naturally, skin color or kam hoga, it would be very fair. And 0, of course, we know represents the albino situation. Now let's quickly move on and try to do a hypothetical cross. When one of my parents is going to be of the darkest skin type and the other is going to be of the lightest or albino skin. So as discussed, we have moved on to a cross between a parent having completely dominant alleles and a parent having completely recessive alleles. Now let's try to form the gametes. We know that when 3 genes are involved, 8 different gametes are formed. We'll look at that. If you don't get the logic, we'll look at that in a moment. But we know here, in this case, in the parents, only one type of gamete will be formed. 8 gametes will be formed, but all of the same type, having the same genotype. Why? Because it is a homozygous dominant condition for all the 3 genes. Now what we do is we perform a cross, and in the F1 generation, the genotype obtained will be capital A small a, capital B small b, capital C small c. Now in the next step, what we do is we perform a cross between the F1 offsprings. But for that, we need to make the gametes from this given genotype. So this is our aim. I'll give you a very simple way of doing it. What you do is first you consider one gene, let's say of the A type. Write down both the alleles for the gene, capital A and small a. Now come to the second gene, that is the B1. Write down both the alleles in both the cases. Okay. Now come to the third gene and again list down both the alleles in both the cases. I just require some more space here. Okay. So now I do this for the C gene. So I had one capital C, small c. Okay, capital C, small c and capital C, small c. So now what I'll do is simply draw lines and write down the genotype. Let's see. So first would be capital A, capital B, capital C. Second would be capital A, capital B but small c and so on. I'll show you one more, capital A, small b and capital C. Capital A, small b and small c. Similarly, I'm just going to list down the others. realize that when I said 8 different gametes would be formed, here we see 8 different combinations of gametes were formed. Now what I do is, now that I know the gametes, I'm going to perform a cross between the F1. But would you imagine that if I had 8 gametes on one side and 8 gametes on the other side of the Punnett square, I'd end up having 64 squares. Well, let me do that for you. Well, after a lot of effort, I've finally drawn this Punnett square for you. I've already taught you how to draw a Punnett square, so I'll just move ahead. So this is my Punnett square. It has 64 squares. 8 gametes have been listed down on both the sides, so I've made the job easy for you. Now, should I start just filling in? मैं ABC अगर ABC लिखने बैठूंगी, it's going to take me another 1 hour to fill this table. So let me give you a shortcut hack for this. What I'm going to do is just list down the number of dominant genes that I have. Why am I doing it? The number of dominant alleles I already know are going to give me what phenotype am I getting. And that is what I want to do in this case. So I'll simply count the number of dominant alleles and fill that in the box. But this is only valid, mind you, it is only valid in the case of polygenic inheritance. Please do not use this shortcut in the case of tri-hybrid crosses, because the ratio we are going to get here is going to be completely different from that of the tri-hybrid cross. So, disclaimer, this hack is only for polygenic inheritance. So let's begin and see how this works. Now in the first box, I see that I have dominant alleles on both sides. So I'll just write that I have 6 dominant alleles in this case. In the second case, I see that I have 3 dominant alleles here, 2 here. So 5. In the next case, it's going to be 3 here and 2 here, again 5. Let's come to, let's say, the last box. I have small a, small b, small c, again small a, small b, small c. So I have 0 alleles of the dominant type. I'll go ahead and fill in the boxes. Well, I've listed down the number of dominant alleles for you. So after filling out this table should look something like this. Now what is the advantage of doing this? Well, I'll just go on and keep counting. Let's say 6 dominant alleles. How many times does the number 6 appear? Let's count. So we'll see, I get 6 only over here, only once. So I'll write 1 time. Let's say 5 dominant alleles, कितनी बार दिखते हैं? I'd recommend going row wise. 1, 2, 3, then here I get 4, then I get 5, then I don't see here, okay, and then 6, don't see here, okay. So well, it appears 6 times. I'll continue the counting, I'll just write down the numbers. Well, what is the advantage of writing down such numbers? The clear advantage is that I'll be able to derive a phenotypic ratio for my polygenic inheritance. The phenotypic ratio comes as 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. यहाँ तक forward जाइए, then just go reverse. You'd get the ratio. It's easy to remember as well. So this was the ratio for my polygenic inheritance. Let's quickly move on to the last part and try to get a graph for the polygenic inheritance. Well, in attempt to draw the graph for polygenic inheritance, what will we write on both the axes? On the y-axis is going to be percent proportion of the population. What proportion of the population lies in which skin color type? So on the x-axis, I will have the skin color type, the spectrum of skin colors. On the y-axis, I am going to write the proportion of population. So my population was how much? It was 64. So my aim is कि 64 individuals में कितने individuals, let's say, of the albino type? I know the answer is 1. So I'm just going to go ahead and plot it on the graph. I know one individual has the black skin color out of 64. So I'm going to plot 1 somewhere around here. Now very dark brown, I know 6 individuals are of this type. So 6 would be somewhere around here. I'll just go ahead and keep doing this. 15 would be here, then 20 is here, again 15 lies here, then again 6 and 1. Now, if I join these points, I get what we call the famous bell-shaped curve. The famous bell-shaped curve for polygenic inheritance. Well, this polygenic inheritance in man was studied by Sir Francis Galton. Sir Francis Galton in 1883. So this is not something which is very new to us. These studies were done a long time back. So this was all for polygenic inheritance. In the next video, we'll start talking about another phenomena of genetics. Do consider subscribing to the channel. If you're interested in watching a video on tri-hybrid cross, do write in the comment section and let me know. And make a separate video on tri-hybrid cross and tell you about the phenotypic and genotypic ratios in that case. We'd also do a hack on how to make a forked line diagram to make the tri-hybrid cross easy. Do let me know and don't forget to subscribe to my channel. Thank you.